this is Joshua Gong from Agriculture and Agri-Food Canada in Guelph. Thanks for this opportunity to present our research. My talk today is about a novel PilRS two-component system that regulates any pilus production in Clostridium perfringens. Dr. Diane has given a talk yesterday about the Clostridium perfringens pilus, and he has given a lot of background. So it saved me a lot of time. So briefly, necrotic enteritis is an important endemic disease in chickens, which costs about $6 billion per year worldwide. This disease has been controlled by antibiotics in feed in the past. Necrotic enteritis is caused by type G Clostridium perfringens, which produces NAB protein, NAB toxin. There are multiple factors contributing to any development, including Agr-like quorum-sensing, dependent pilus. This slide shows the Agr-like quorum-sensing in Clostridium perfringens. There are four major components called AGRB and D, VirSR and S. AGRB and VirSS are located on the cell membrane, and AGRD and VirSR are located inside of cells. AGRD is responsible for production of the precursor of an auto-inducing peptide called AIP. AIP is transported to outside of cells through the AGRB and becomes mature. Matured AIP interact with the VirSS, which is in turn interact with the VirSR to achieve the regulation of transcription level for gene expression. This system has regulated more than 140 genes in the Clostridium perfringens. In 2013, our group has published a paper about NE pilus locus. In this locus, there are three structural components of the pilin proteins. In addition, the saltase indicated by black color. In the downstream, there are two components labeled with green. They are called PilR and PilS. They appear to be two component regulation systems. However, their functions are unknown. The previous study has revealed that pilus is required for any development. This is done by Robert Moore in Australia, Robert Moore groups in Australia, and our groups. And what's the function of the PilR and the PilRS? In addition, will the production of a pili is regulated by Agr quorum-sensing? To address these questions, we have generated a mutant of the PilR and the complement through using cross-channel technology and also shuttle vector between Clostridium perfringens and E. coli. We also took advantage that we have the mutants of the VirSR and the pilin and the pilus. So by comparing the mutants and complement with the wild type for their behavior, we will know what's exactly going on there. And this slide shows the Western blot analysis. There are four panels. They are identical. Panel A is Comex blue staining. B, C, and D are Western blot probed by antibody against CNA, fin A, and fin B, respectively. I just want to focus on panel B. So on the first line on left, is the wild type CP1, and that's the production of the CNA protein. And next three lanes, that shows near non-production of the CNA protein. And the next two names, one is the Agr mutant, the other one is the complement. So the production of the CNA protein is increased or in the mutants. However, in the complement, the production of the CNA protein was significantly reduced compared to the wild type. The next two names shows the results of the VirSR mutants and the complement. The same situation happened. The VirSR mutants also shows similar level of production, or a little bit increased the production of CNA protein, but in the complement, the amount of CNA protein reduced. The next two names shows the results of the PilR mutants and the complement. The PilR mutant produced almost none CNA protein, but in the complement, the production of CNA was significantly increased compared to the wild type. So the same situation and the pattern have been observed for antibody against the film A and the film B in the Western blot. So it appears that the PilR positively regulate the production of PilI proteins. However, quorum-sensing system negatively regulate the production of the PilI proteins. So there's another question people can ask: would be the production of the Agr regulate system regulated by something else? So then we need to check the function. To confirm the function, so we use the binding experiment. So this slide shows 4 panels. A, B, C, and D shows the binding of the PilR mutant and the component and the complement. Agr mutants and the complement. VirSR mutants and the complement. And pilus mutants, respectively. So there are 5 types of collagen were used. So indicated here in type 1, 2, 3, 4, and 5. So in each panel, black bar shows present the white type, while the dark green bar represents the mutant. The light gray bar represents the complement. So let's focus on the panel A first. So compare the wild type and the mutant. The mutant binding to the collagen was significantly reduced. However, when you compare the binding of the wild type with the complement, they were in a similar level. And this, when you look at the binding to different types of the collagens, the similar trends were also observed. Then in panel B is binding of the mutants of Agr and the complement to different types of the collagens. The situation was opposite. When compare the wild type and the mutants, this dark and the dark, this black bar and the dark gray, so there are no significant difference in binding. However, compared to the complement, the binding of complement was significantly reduced. And this situation was also across the binding to different type types of collagens. And similar pattern was observed for the VirSR mutants and its complement to binding different types of collagens. In panel D, all the mutants of the PilRS showed significantly reduced binding to all types of collagen. So the question is, the increased binding in the mutants of the Agrb and the VirSRb is specific or due to there's another factor which is responsible for the binding. To address this question, we took an approach to examine the blocking of the binding by different antibodies against different PilI protein at a different concentration. If the antibody shows blocking effect in a dose response manner, so which would suggest that the binding is mediated by the PilRS and the binding is specific. Otherwise, there could be another component which are responsible for the binding of a mutant to the collagen. So in this slide, the panel A and the B are blocked by the antibody against the CNA at a different concentration, while panel C and the B blocked by the antibody against the fin A. In each panel, we compare the behavior of the wild type CP1 and the mutant Agrb. As you can tell from the slide, they all show dose response to when the antibody concentration decreased, the binding increased. So this suggests that the binding of the mutant or wild type to the collagens is mediated by PilRS specifically. So this leads to a conclusion. The first one, PilRS two-component system positively regulates pilin production, while Agr-like quorum-sensing appears to negatively regulate the pilin production in Clostridium perfringens. By this point, so what can be done in the future? There are some questions still remain. So how's the regulation of PilI by PilR and S in vivo? And the second question, what else are regulated by PilR and S, this new two-component system? The third one, which part of CNA is responsible for the endogenicity or blocking of the adhesion by the antibody? This would present some chance to improve the seed vaccine and for other opportunities. Finally, I would like to acknowledge those people in the slides, the Dion Lip, Yuan Zhu, and Jason Carroll, and Hai Yu, and Chimbo Yang, and we also very appreciate the funding from CPRC and Agriculture and Agri-Food Canada. This project is actually funded through the Poultry Science Cluster 3. Thank you.
Thank you, Dr. Gong, for your presentation, which is really very interesting. I was wondering if you could develop a bit more on the possible immunogenicity of your pilin system. Is this something that you're looking into?
Yes, we are going to develop a product. Actually, we have reached agreement with a multi-international company for product development. So we're going to develop a vaccine against necrotic enteritis using the PilI protein.
Great. Well, this sounds really exciting. And
we also got one graduate student to start in 4. If everything goes well, and he may do some crystallization of the CNA.
Oh, so that will give us really a visualization of the system.
This would depend on how lucky we'll be, though.
Well, best of luck and congratulations for this very interesting work.